Yeah, thanks. 1 second, I'll start sharing. Okay, can you see this
Yep.
slide? Fine. Okay. Yeah, as Ben said, my name is Zach Crockett. I'm at Oak Ridge National Lab, and I'm going to be talking about publishing data and narratives with KBase specifically, and a little bit about, in general, what we are trying to promote in the culture of data publishing. So first, I'll talk about just the general concept of data publication and citation and how data is tracked in KBase. And then I'll go into some examples and an overview of how you can publish your data through narratives. And so first, I want to talk about why you would publish data. Historically, data has been just part of publications, papers about other, as a small part of a paper. But the data itself is often a scientific product in its own merit, and is something that you can use outside of the paper that it came with, and that should be shared with others, and where credit is maintained. And a big part of that is maintaining PIDs, which are persistent identifiers, to keep track of the data. And you probably would recognize those most as DOIs, or digital object identifiers, which provide a unique identification of a data set or paper. And so we at KBase have worked with the Department of Energy's Offices of Scientific and Technical Information to create a form of DOIs for data sets specifically. And we can assign those to your narratives if you publish them. And we're also working with other organizations and publishers to improve data citation culture. Some of the main ones are ORCID, as I mentioned, EMSL, which, if you don't know, is the Environmental Molecular Sciences Laboratory, NMDC, the National Microbiome Data Collaborative, and more. And this is, data in general is a growing concept in all fields. Google has recently, within the past couple of years, added a data set search within as a complement to their Google Scholar search that goes beyond just Scholar into the data sets themselves. And you can find some of our published narratives in this Google dataset search already. And so data in KBase, a lot of the relations between data in KBase is not something that people normally pay attention to, because it's a little bit behind the scenes. But all of when you run an app in KBase or upload data, all of the processes that lead to the creation of that piece of data are tracked through the system. And you can follow that chain of relations through all the apps, all the instances of sharing or copying. You can follow the path that data takes from when it's uploaded to when it's transformed into other objects. And so I'm going to stop sharing this slide for a second and show an example of that. So I have these two narratives. One of these was a public narrative created by a user here at Oak Ridge, Dale Pelletier, as part of the publication. And so I've copied that narrative to do some text updates on my own. And so if you follow this object, you see the object's permanent ID is here, and that the prefix of that is the same as the narrative that it's in. But if I look at this data provenance map, you can see the relations between the objects that led to that. So this object is the object in my narrative, and it is a FBA model that was created from these other objects. Another FBA model and actually 2 FBA models from Dale's original narrative. And then you can follow that chain. So for instance, if I follow this object to its landing page, I can see the same information for this object. And you can see the reactions that come from this and the reports and the genome that was used to build it. And again, I can follow that all the way back to when Dale uploaded the original sequence data for that. And so that allows you to track the data, and it makes the data more reproducible and easier to follow. And so now I'll move into the basics of the narrative itself. So presumably most everyone on here is a KBase user and has at least opened a narrative. But if you haven't, there's a few main features of the narrative: the data panel, the apps panel, and then the main body of the narrative, which contains markdown app cells, code cells. And I've highlighted in blue and yellow the parts that will be shown if you create a static narrative, which we'll see in a minute. So the important things to see are the data objects are the data set, and then the cells in the narrative are what are used to generate and annotate the analysis. And so to answer the question that was put in the chat, so when you first create a narrative, everything is private. And then you can make your narrative public to share it with everyone, or you can share it with specific users. And then when it's made public, you can create a static narrative from it. And so a static narrative can be generated for any public narrative in KBase. And a static narrative is a snapshot of the narrative at the time of creation for that particular version. And all of static narratives are visible outside the KBase login. And I can show an example of one right now. So this is the static narrative version of that narrative I showed just a second ago. So all of the markdown cells are rendered just as they appear in the live narrative, and the app cells and result cells appear the same. For the most part, you'll see some, most apps that have a visualization or report will show up here, but some things like this assembly, you'd need to actually view the live narrative to see all of the features of that assembly. And so there are a lot of times some misconceptions about static narratives when I've found when I talk to people about them. So what they do and don't do is sometimes a little bit unclear. And what they do is to create a record of a single version of a narrative that is separate from the narrative itself. And they allow the viewing of most parts of the narrative outside of the login. And it packages most of those app cells and results into a static image or representation. And what they don't do is lock the underlying narrative. So when we say you make a static narrative, it doesn't mean that you freeze the narrative in place. The underlying narrative can be changed indefinitely. And those changes will not be reflected in the static narrative that you create, but you can create a new version of the static narrative, which shows the changes that you've added. And both the new and the old version of the static narrative will still exist. And static narratives don't allow immediate access to download all the data. So when you create a KBase object, it's stored on KBase servers. And to download it, if you wanted to download the genome, for instance, as a GenBank file, it would need to run an exporter app. So that functionality is still behind the login. But if someone wanted to download your data, they could simply log in and download it through that. And so now I'm going to talk for a bit about some tips for improving narratives beyond just having app cells in your analysis. And so the key to making a high quality polished narrative is to use markdown cells liberally. In the example that I showed, we had a cell for the introduction to the narrative, which often I advise people just to copy and paste the abstract from the paper it's associated with. And then you can include a table of contents and an explanation for each step. You don't have to necessarily provide an explanation for every cell, but if you have several assemblies that you annotate, it would be helpful just to explain that you annotated these assemblies using these parameters and why you chose those parameters if they're outside the default settings. And I've put these slides in the Q&A doc and included some handy cheat sheets that I use for Markdown and HTML formatting, and also a narrative that you can copy into your narratives and copy and paste the formatting into any Markdown cells you want to include. And so a couple of quick things that I find are the most helpful and the best return for your work when it comes to organizing and annotating narratives are a table of contents and pictures and graphs. So the table of contents, well, for one, it allows people to see what's in your narrative from the start. And also you can combine a HTML list with an HTML link and hyperlink within the narrative to other sections of the narrative. And I included that in this narrative. And in my copy, I can show you the HTML I used to generate it. So I have this list for table of contents, and in the list, I've put the HTML A tag on it to refer to different steps of the analysis. And so when you click on this link, it will take you to that section of the narrative, although this link is broken. This link works. And so you see the name of that link is added to the URL, and it redirects to this section of the narrative, which it comes in handy when you have a large narrative with dozens or more app cells. It can be tedious to have to scroll through that to find a particular section. And so now I'll show a few examples of scientific workflows that you can do in KBase, and you can share as a public or static narrative. And so there's a few examples. So the most common one would be this first example, where you just have a straightforward analysis. You do a handful of steps of apps, and then you publish the results. Then there's other versions that can happen. For instance, the second type, you might possibly do two different parallel analyses, which can then be combined or linked from the same README narrative, which just links to those analyses. And then that narrative, the landing page narrative, is used in a publication. And then another version might be a template narrative, where you do, if you're doing the same type of analysis repeatedly, you may create one and then run the same steps on different sets of data, and then either publish those separately or as one collection. And then you could also do a narrative where you do analysis and then publish that analysis, and then you later come back and want to build on that. and you continue the narrative with more steps, and then you publish that. And you could make a static narrative for each step. Like in this instance, you can make a static narrative at step 3 and at step 6 and publish both of those. And so I've got some examples of some of these. In this first one, the narrative I've been showing is kind of an example of that first set of just one straight through analysis. And it's a little bit similar to the template model where the plant microbe interfaces group at Oak Ridge, Dale and Mircea Podar have been putting out a lot of genome announcements. And we've created narratives with them for all of those. And so they follow the same general template for the most part. There's a little bit different analysis, but they keep all the information. The experimental methods are included in a markdown cell, and then the steps of the analysis in KBase are included and annotated. And then we created a static narrative for that, and it links to the genome announcement for all of these. And then another example of that second set is a paper from Andrew St. James at Cornell University. So he did a lot of analysis on KBase in several narratives with dozens of objects. And then he published just his general analysis in KBase. And so we worked with him to combine some of that into this parent narrative, which can be seen here. So this narrative, if you look at the data tab, only has the mags. These are the genomes that he included in his publication. But this narrative, it links to his metagenomic analysis and his metatranscriptomic analysis. And both of those have their own narratives that you can see if you follow the links. And so these contain a lot more data. So not all of this data was used in his paper. A lot of it was just exploratory. You see he's got done some species trees that I don't believe are included in the final publication. And some of these analysis steps were repeated for multiple sets of data, but this provides just one example. And then in a sort of supplementary data at the bottom, he's included all the files. So this just this narrative just includes all of the files across all of his analysis that were used in that paper. And so when we create a PID for this analysis, this data set in KBase, we do it only for the landing page for this narrative. And then that links to all the other narratives. So the DOI for this data encompasses all of it through this one sort of portal narrative. And then another example of a sort of workflow narrative is this Delftia Narratives created by Rose Krebs, who's a student at North Carolina State. And she created this template narrative, which has the steps to do, but they've not been done. So she put the app cells in place with the explanation for how to use them and what you'd be doing in this narrative. And there is nothing in this. So when she does analysis with it, I believe this is part of an assignment. So when other students use this, they create their own, which you could see in a narrative like this one. And so this has the same information. It's copied from that previous narrative. And this one actually has some data in it that you can see. And so if you were doing these as a publication to get an ID in KBase, you wouldn't get a DOI for that first one, but you could get a DOI for all of the subsequent narratives. And then an example, a recent example that we've just worked with an author to do is for a non-KBase user. So this paper, they did a metagenomic analysis from some ocean samples. And one of our educators in the KBase educators org wanted to use this data for a class. And so I worked with the author to upload the data. And I believe in the paper, they assembled it with either MegaHit or Proca, and annotated with Proca. And so all of the assemblies are uploaded. All the metagenome assembled genomes are uploaded, as well as the metagenomic reads. So the students in that class could reproduce that analysis to see if to make sure that they get the same results that the authors got in their paper. And then if they perform any further analysis that's worthy of a publication, then that can be cited and provide credit for this author who didn't do the analysis in KBase. But we maintain the credit being applied to her publication. And so that's something if you do want to work with data from another publication, then you can have the owner of that data contact us, and we can work with them to get their data in the narrative and make sure that they are maintained as the author and not someone in KBase. And so on that note, we want to work with you, the users, to get your data published and publicized. And so as I mentioned, we can register your narratives as datasets with DOIs. And this can be tied to ORCID accounts. You can manually add those DOIs to your ORCID if you have an ORCID account. And we hope to, with future development, make that process automatic and have DOIs generated by KBase immediately sent to your ORCID account. But that would be some future work. And then if you do publish with KBase, we'd be happy to add your publications to the highlight page on the KBase website and advertise on Twitter and more. And so if you do have a narrative that you want to polish and publish, then you can contact us at engage@KBase. And so I want to leave some time to talk about our next steps and get some and send out sort of a request to you, the users, for what you would want to see. So as we develop this further, this static narratives are sort of in a 1.0 state, and we're working on expanding the functionality. One of the biggest things is to make the version history a little bit more accessible. As I mentioned, all of the versions still exist, but we don't currently have an easy way to see them all at once. And we want to improve the visible author control. If you, for example, on this narrative, because I edited this narrative and worked with Dale, I'm listed as one of the narrative authors and editors. And so we want to improve the manual control of that. And then we also want to make the process for getting a DOI more automatic. As it is now, it's a manual web form that I fill out. So I have to email back and forth with the authors. And so we want to get that more streamlined and more automatic. And so in that note, we want to hear from you for how you would want to do that. How would you expect to be able to publish a DOI? There are a lot of fields that would we keep track of in KBase, like in your profile page. You can put your affiliation and your funding source, but I don't know if people do that, or how often you update your profile. And so if we pull from that automatically, that could create issues for generating the DOI with the wrong affiliations. And then beyond that, we want to work with the data community and the scientific community to improve the culture and encourage people to make data sets a thing worth citing in their own right, beyond just being crammed in the supplementary information for a paper, or even worse, just mentioned and not included in the paper metadata. And so any ideas or questions you have, you can always send to us at Engage, or you can email me directly. My email's in that Q&A doc. And that's all I have to talk about today. I can take time for questions or ideas if you have any. I see somebody is typing in the document now. And yeah, so the question is, when we're using a narrative to analyze new data, how does the previous data and results get deleted? So you don't delete data. So if you were to, I'll make a copy of this narrative just to show. So if I do create a new narrative copied from this one, the old data will still exist. It won't affect the narrative it was copied from. And so if I were to delete this object, I could delete this object. I could delete the version for narrative 87138, and that one would be gone, but it wouldn't affect the underlying data. And so it would just remove the linkage to the underlying data in this narrative. And the same with app cells. We don't encourage you to delete them because it removes, it makes it harder to see the connections in the data. They're still there and you can search for them. It just makes them less intuitive and more difficult to follow. So you can delete a cell if it's an error, or if you decide not to run anything from it. And then the question is, how do you use that copied narrative for new analysis then if the original data is not deleted? So you could, if you were to add data, so in this case, if you wanted to add a new assembly, you could just import a new object. If you wanted to reproduce this and add another genome for each step, then you could just add you could import another assembly and then add the same app cell. So you can go to the search panel and just add another annotate microbial assembly. And then you just add your new objects in parallel with your old object. Or if you want to start from scratch and just use one as a template, you could just you could view the narrative you want to copy, and then open a new narrative and add all the steps in a fresh narrative.
Right. You can also create a template narrative with just empty apps and save.
Yeah, yes, I can show that one again. So this one, let me show you what it looks like in the narrative environment. So this is the template app that Rose created. And so the data panel is empty and the apps are all fresh and unrun. So if you were wanting to reproduce this, the best thing to do here would just be to make another copy where you maintain the template. And then when that opens, then you can add your own data or new data if it's, and just, you know, rename it to make it more recognizable. And the question, do all narratives have that option to be opened as a template? So for any narratives that aren't yours, that they will only be only have the option to be copied. So if you just view any public narrative and open it, you can only, one second while this loads. Yeah, you can only copy it. So you can't add your own, I can't add my own to this because it's in view only mode, but I can copy it. So to make it a template is sort of up to the original author to include the word template or just any indication that's a template in the name. And from there, you can treat that narrative as a template. So the template narrative is not really an official KBase thing. It's just sort of an extension of how narratives work. And someone asked if a new app can be developed, and Alicia's answering that, but yes, KBase is open source. So any apps that you have that you want to add to KBase, you can work with us to add into the KBase environment. Or you could, if there's an app you want to see in KBase, you can ask the author to work with us.
All right, thank you, Zach. Not seeing any more questions. Of course, like Zach said, if you want to follow up or you have any interest in helping or allowing us to help you promote and publish your work that you're doing in KBase, please reach out to us at the engage@kbase.us email account, or you can follow us up on Twitter or YouTube, etc. Just reach out to us and let us know how we can help. And of course, just one other thing is that we do try to post all of the new publications on our website, you know, regardless if we're promoting them or not. Yeah, we do link to publications with narratives in it. So we just would encourage you to put these narratives in your publications as you're publishing your research, so that you know, we can really help out with the reproducibility mission that we have as part of our mission as KBase. So with that,
Can I add one more thing, Ben?
yeah, go for
in
it.
relation to that, Alicia especially, but a lot of us at KBase are working to make this data more integral to publications. And so if you are publishing and your journal does not have a data availability statement, you can contact Engage or me directly, and we can get in touch with the author or the publisher, because we really want to push that.
All right, thanks everyone, and we'll see you on the next webinar.